Okay, well, let's get started here. I'd just like to welcome everyone to our fourth seminar in the Geomicrobiology Network UK seminar series. I'm John Moreau, and my co-host is here, Dr. Karen Cameron. We're at the University of Glasgow, and we'd just like to quickly thank our sponsor, Mineralogical Society of the UK, and specifically its executive director, Kevin Murphy, they've been gracious to support our series thus far this term, and it's going well. We're glad to have you all here. We've had a great turnout so far, and today seems to be a continuation of that, so that's great, because we're really excited today to have Dr. Olga Golishina from Bangor University. She is a reader there in the Center for Environmental Technology and a specialist in acidic microbiology, acidic ecosystems and microbiomes. We'll get to hear about Archaea today, which is something I think we haven't heard about yet in our seminar series, so that's quite exciting. From extreme environments, acidic environments, and cold temperature environments. And at the center, they work on things ranging from microbial ecology in Olga's group to novel enzyme biotechnology. So that's a really exciting mix of basic and applied research. Without further ado, we'll turn it over to Olga. I'll just ask that you hold any questions to the end unless there are points of clarification, in which case she's quite happy for you to chime in and ask a question during the talk if you like. And I'd just like to note that we are recording, so if you want to keep your microphone and camera off, that's absolutely fine. So without further ado, I'll turn it over to Olga and her talk today, the Acidophilic Archaea from environments with low to moderate temperatures. Thank you, Olga.
Okay, thank you very much, John, for this introduction, and thank you very much for invitation to participate in this seminar series. And today I will speak about acidophilic archaea from environments with low to moderate temperature, or by other words, from non-thermal environments. And actually, what environments are considered to be acidic ones, and what factors are necessary for the generation of these conditions. Firstly, it should be the presence of reduced sulfur compound at quite high abundance. Then conditions should be conductive to the microbial oxidation of the reduced sulfur compounds, as for example, presence of oxygen. And the third factor, it's actually acidity needs to exceed neutralizing capacity of the environment. And what environments could be qualified to be low pH environments? Firstly, it's environments which are actually connected with geothermal activities, like volcanic environments, terrestrial, or for example, marine ones, and also cave systems, acid sulfate soils, and mine impacted environments like shown here onto these pictures, like Rio Tinto, which is natural environment, and also Paris Mountain shown on the picture here. And metal or bodies are actually places of concentration of sulfide minerals, and pyrite is the most ubiquitous of all sulfur minerals. And the oxidative dissolution of pyrite by acidophilic microorganisms and its role in generation of acidic metal-rich pollution, such as acid mine drainage, or shortly AMD, is among the most intensively studied processes in acidophile microbiology. You could see on this map that AMD emplacements are globally represented and were studied with different level of details. You could see that pH in all these environments normally less than 3, and temperature has some variety, but normally it's less than 30, with Paris Mountain average temperature about 8 9. Probably this site located on Anglesey is the most characterized with the most low temperature. You could also see from this table that this actually emplacement have very high concentrations of heavy metals. Organisms inhabiting these sites are represented by eukarya, bacteria, and archaea. And today we will mostly speak about archaea with you. And organisms represented by this group are mostly Archaeota, but also some filterable archaea and some other group which we will discuss today. As you could see from this diagram, with decreasing of pH, we could observe increase in archaeal numbers. Normally at pH less than 2, archaea could increase their abundance in this environment. And the very first group of archaea which we will discuss today is the order Thermoplasmatalis of the class Thermoplasmata, phylum Archaeota. But please be aware that nowadays happening a lot of changes with classification and new taxonomy, and according to new GTDB classification, this phylum where belong order Thermoplasmatalis and class Thermoplasmata belongs now to the new phylum Thermoplasmatota. It still sounds quite unusual for us, but we all need to be used to this. And these Thermoplasmatalis organisms are the most acidophilic prokaryotic organisms known so far. They have very low pH optima and pH range. They are mostly growing at pH lower than 2, and some strains are also able to grow at pH 0, for example, Acidiplasma, Ferroplasma, as well Cuniculiplasma, and Picrophilus is also able to grow at negative values of pH. And the second feature which makes this group very unusual, very unique, it's their pleomorphic morphology, which is consequence of the lack of cell wall. And please note that this is the only archaeal group which shows the lack of cell walls. Right. So what we know about organisms within this order Thermoplasmatalis? Here on this phylogenetic tree, you could see that currently 6 genera are included into this order. Thermoplasma, which was described back in 1970 as Mycoplasma from coal refused file. Then it's Picrophilus, Ferroplasma, Acidiplasma, Thermogymnemonas, and the very last member which joined this club was Cuniculiplasma divulgatum. Please note that only sequences of organisms with validly published names are included into this phylogenetic tree. If we will look on general overview of features of all these archaea, you could see that these features are pretty similar in terms of cell morphology. The only exception is Picrophilus, which has S layer, and because of this, it has irregular cocci shape. Also, these organisms have mostly organoheterotrophic lifestyle, but some members like Ferroplasma and Acidiplasma are also able to iron oxidation and reduction. And if you will see on this range for temperature, you could see that these organisms could be divided on moderate thermophiles and also on mesophiles. And you could see that actually this range is quite wide, but optimal temperature are normally in mesophilic range. Furthermore, all these organisms are very acidophilic, and some of them are aerobic or facultative anaerobic ones. There is also huge diversity among uncultured archaea within the orthothermoplasmatalis. And here it's the example of only one site, which is Iron Mountain in US. And you could see that there is a lot of different cluster of these organisms, which were given name alphabet plasma, represented within the order Thermoplasmatalis. And actually what we also could see that this cluster G plasma is 100% identical on 16S rRNA gene with Cuniculiplasma divulgatum. So from where these last strains of the order Thermoplasmatalis Cuniculiplasma divulgatum were isolated, both these strains were isolated from copper mines located on Anglesey in the UK, and also from southwest of Spain, which could be seen on this map here. And Cuniculiplasma is actually G plasma, and earlier these G plasma sequences were shown to be the third abundant community members in Iron Mountain metagenomes, contributing up to 22% of total community proteome. And another place, Cryosphere system, also showed very high representation of Cuniculiplasma, about 15% of total metagenomic reads in metagenome. Right. Our analysis of metagenomic data across the globe showed that Cuniculiplasma, like organisms, are represented widely in acid mine drainage places, and also we have discovered the presence of Cuniculiplasma-like organisms from high Arctic, from actually Svalbard samples, and Cuniculiplasma is also able to populate geothermal environments. What is the physiology of Cuniculiplasmas and of all Thermoplasmatalis in these types of environments? They are mostly organotrophs, and they're using organic material in natural habitats coming from dead cells or from exudates of metabolizing or dead cells. So they're scavengers, mostly scavengers. And we have also observed quite substantial set of genes encoding proteolytic enzymes in genomes of Cuniculiplasma, Ferroplasma, and other Thermoplasmatalis. And in laboratory, all these organic compounds could be substituted by different polypeptides. What actually lesson we got from the comparison of genomes of two strains of Cuniculiplasma and of metagenomic assembly G plasma from Iron Mountain? Despite the difference between places of isolation of all these organisms, they have pretty similar genome sizes, and also certain differences could be seen here on average amino acids identity of core proteins could be seen here in the center of this picture. Furthermore, altogether, many cuniculoplasma-like sequences exist now in databases. And you could see from this picture on the right side that all these genomes of different organisms isolated from different IMD sites across the globe are quite similar between each other. We have also observed quite interesting feature, which is actually the similar placement of genomic islands in genomes of two these strains from Paris Mountain from UK and also of Spanish one. And please also pay attention that they have different number of genomic islands. Paris Mountain strain has 4 genomic islands and Spanish has 10. And this genomic island incorporate mostly genes of toxin, antitoxin systems, restriction modification systems, also associated with metal efflux and oxidative stress response. We have also observed in genomes of cuniculoplasma so-called copper fitness island, which probably quite important for these organisms for the survival strategy in acidic settings with quite high concentrations of heavy metals. You could see that the central position is actually belongs to cation transport ATPases, and the actually placement and all those genes are mostly belonging to certain Archaea families, and you could see that the position is quite similar in different genomes. What we also know from the analysis of thermoplasma and of thermoplasmatalis and of cuniculoplasma particularly, that these organisms have a number of genes which help them to cope with high concentrations of heavy metals. They have metallochaperones genes, heavy metal reductases, mercury 2 reductases, and also Ars-A, B, and R family proteins, which are also quite important for heavy metal resistance mechanism. Right. So just to understand actually what helps this organism like cuniculoplasma divulgatum, which is actually mesophile, to occupy some low temperature niches, such as, for example, Paris Mountain, where the average average year temperature is only 8-9 degrees, or for example, high Arctic places. We did a proteomic study of cuniculoplasma divulgatum in control conditions, 37 degrees, and also in cold conditions. For this, we put organisms for 3 hours at 0 degrees, and we observed quite high increase in expression in low abundance proteins. What actually were these proteins? It was proteins like metal-dependent hydrolase used for polypeptide hydrolysis, or for fat-dependent oxidoreductase for oxidation of amino acids. Also, proteins involved in central metabolic pathways like aspartate carbamyltransferase, regulatory chain proteins involved in pyrimidine biosynthesis, or 2-oxoacid ferredoxin oxidoreductase involved in citrate cycle, or V-type ATP synthase involved in ATP production. We have also observed a substantial increase up to 6% in beta subunit of thermosome and also of glutamate dehydrogenase. And glutamate dehydrogenase was increased up to 9%. And these enzymes could be used in deamination of glutamate originated from polypeptides. Furthermore, we have observed certain increase in proteins involved in cysteine, methionine, and glycine metabolism. And actually, our suggestion that cysteine could be crucial for disulfide bonds formation and for generation of pyruvate. Furthermore, we observed decrease in methionine, and we expect that overexpression of methionine could be important for the synthesis of some of S-adenosyl methionine, and some synthesize is one of the most expressed proteins which provides important methyl donor groups for methylation of DNA, for example. Furthermore, we consider that methionine and cysteine are important for the production of cofactors, right? And also could be quite important for production of cobalamin, folic acid, or for methylation of actually various compounds. And cysteine synthase could also catalyze a bidirectional reaction between cysteine and serine as well. So, and we have also found increase in glycine uptake system. Very interesting feature for which we discovered for cuniculiplasma. It was the coexistence of this organism during the isolation with microarchaea or Arman2-like organisms in vitro and in situ. And you could see on this picture on the right side that actually we found some clusters of contigs in metagenomic data related to the presence of cuniculiplasma and certain type of microarchaea near 14, and we have also found clustering for some other group of thermoplasmatalis with other microarchaea in metagenomic data. Just to remind you what is this group, and this Arman or archaeal Richmond mine acidophilic nanoorganism was firstly discovered by Bamfeld Group in 2006 in Iron Mountain in US, and the name Microarchaea and Parvaarchaea, two new phylum names were given to this organism. And nowadays, Microarchaea is still considered to be the level of phylum according to the new GTDB classification. But the status of Parvaarchaea is now actually moved to be the order of Parvaarchaealis within the phylum Nanoarchaea. And Microarchaea and Parvaarchaea is the second group of archaea which are found in acid mine drainage environments and in other acidic environments as well. And if thermoplasmatalis could be found as a major group in this environment, Arman or Microarchaea, Parvaarchaea are normally represented as minor group only in this data, but there are some exceptions when Microarchaea or Parvaarchaea could be represented in rather higher numbers. What was very interesting that these organisms were actually found in filterable fraction, and the cells were considered to be of very small size, something about 0.15, 0.2 micron only, and genomes of different level of completeness recovered from metagenomes also revealed that these organisms have very, very small genomes, less than actually 1 mega base. And actually, all these features are quite characteristics. I mean, small cell sizes and small genomes for small archaeal organisms, which could be host-associated or which could be symbiotic microorganisms. Currently, we know that microarchaeota and parvoarchaeota are widely distributed in different acidic environments, including IMD environments and also geothermal environments. And you could see it here that these groups could be divided on different families and on different genera. But quite importantly, that currently there is still very little we know about physiology of this group. So during our work, we were lucky enough to get the complete genome of microarchaeota representative, which we called MIA14, and gave name Candidatus manca archaeum acidiphyllum. And what we actually found that the genome is very small, less than 1 mega, has quite a big proportion of hypothetical proteins, also has 3 introns. We found no genes for many central metabolic processes, like, for example, glycolysis, or also TCA pathway, or synthesis of many amino acids. We also found almost no genes for respiration, and also no genes for the production of vitamins or coenzymes, and so on. And quite importantly, that actually we consider that this incompleteness or absence of many central metabolic pathway and reduced genome size provide evidence that these organisms could be symbiotic, relying on host, and host is very important for the provision or metabolites, energy, and also probably different kind of macro molecules between manca archaeum acidiphyllum and its host, which was in our particular case cuniculi plasma divulgatum. Right, quite interesting was the feature that we compared genomes of 2 different strains of cuniculi plasma and also of MIA14. And we realized that actually there is a set of orthologous genes shared between MIA14 and the Spanish strain cuniculi plasma divulgatum, but these genes were absent in cuniculi plasma divulgatum PM4 strain. And the presence of these genes in MIA14 and absence of these genes in Cuniculiplasma divulgatum actually point on quite close evolution history of these organisms, and that certain genes could be transferred between Thermoplasmatalis and MIA. The set of genes which were found within this lateral gene transfer event were membrane-associated proteins, some dependent methyltransferases, peroxyredoxin, and also sulfocyanine. And the lack of sulfocyanine in MIA14 and the presence of this gene in MIA14 is quite important, because the loss of this gene was quite costly for PM4 strain, which is growing relatively badly in comparison to Spanish strain S5. We have also found 5 genomic islands in MIA14, and the largest genomic island has about 41 genes, with about 15 of these genes which cannot be associated with any ARCOX currently. Right, and we also compared the genomes of MIA14s with other phylogenetically related organisms, which could be seen on this phylogenetic tree. And actually what we discovered here, that in comparison to other organisms belonging to TPAN supercluster, TPAN, it is Diaferotrita parva Archaeota, Anigma Archaeota, Nana Archaeota, and Nanohala Archaeota, and all these organisms shown here are actually affiliated to TPAN supercluster. MIA14 or Archaeum acidophilum experience unusually high level of gene flux, and gene gains and gene losses seem to affect basically all functional groups equally, with probably with a notable exception of cell motility group, where more gains than losses were predicted by us. Right. So how diverse are microarchaeota? They are quite diverse, and actually they could be quite diverse even in one place, like for example in Paris Mountain, where we discovered in our metagenomic data 5 different phylotypes of microarchaeota. Another study, which is rather with geothermal environments, revealed that in enrichment culture, microarchaeota could coexist with Cuniculiplasma and Acidiplasma, and also it could be some certain of association of different types of microarchaeota with Cuniculiplasma. And we have a lot of questions here, whether Cuniculiplasma could be the only host for microarchaeota, or whether other organisms could contribute to this association. So we currently don't know. And actually another question, whether Cuniculiplasma could be the host of 2 different types of microarchaeota, we don't know as well. Right. So what we actually learned during our metagenomic work with Paris Mountain microbiome. Contrary to expectations, we found that Archaea could predominate in this low to moderate temperature environment. We found that about 67% of reads were attributed to the phylum Archaea, with 58% of reads which were associated with thermoplasmata, with one particular group of E-plasma actually shown to be in a dominating number. Cuniculoplasma, what is interesting, is represented only minor group here, just 3%. Two other groups which were represented in small numbers as well were also microarchaea and terrestrial miscellaneous euryarchaeotal group or shortly SMEC archaea. So what kind of niches actually association we were able to see? We discovered that archaea are mostly associated with sediments in this environment of acidic streams, and bacteria are mostly associated with water, but there are also some exceptions. For example, microarchaea numbers were low enough everywhere, but it was slight increase in water samples in comparison to sediment samples. We also have found that archaea were represented across all depths in acidic stream environment, with some particular groups represented in higher numbers, like it could be seen here, like this E plasma or B decae variant. Right, the third group, which is represented after thermoplasmatalis and microarchaea and parvoarchaea, it's SMEC, or unclassified thermoplasmata within euryarchaea or thermoplasmata. And basically, currently, we know nothing about this group at all. We just know that they're probably associated with some presence of sulfate, in some cases of methane. And our metagenomic work revealed that these organisms associated with acidic environments are probably represent the new order, which is quite close to the order of Methanamacylococcalis. So another our study of microbial community of polymetallic deposits of acidic ecosystems of continental climatic zone located in Trans-Baikal areas showed that the presence of archaea was relatively small here. And why this environment was actually quite interesting for us, because this environment is characterized by very huge differences in temperature between winter and summer, something between -30 and +22. And all water bodies could be frozen for about 6 months in this environment with actually depth of ice about 1 meter. And pH of 3 environments, which were the focus of our work, were not extremely acidic, something about 2.3 and something about 5. And probably this was the reason why we didn't find many archaea here. It was mostly associated with the site with the most acidic, with the most low pH, like it's shown here, Ulan-Bulag site. And we found again mostly the same group. Here, this SMEC group, the thermoplasmata, and also some microarchaea and taumarchaea, which we will discuss in a minute. And it was also interesting and a good possibility to compare whether there is a difference between organisms populating these unique sites in terms of temperature regime, and to compare organisms inhabiting other acid mine drainage sites from Spain or from UK or from actually US. And quite surprisingly, we found no difference in organisms. Almost the same organisms were found in all these places. Right. So, and finally, the fourth group of Archaea in IMD environment, it could be Taum Archaeota, or according to GTDB classification, it's now class Nitrosospheria, phylum Thermoproteota. And we found some organisms belonging to this phylum in our metagenomic data from Transbaikal area. And also Taum Archaeota were previously shown to inhabit pit lake in Iberian pyrite belt in Spain. And these organisms were mostly associated with pH which is rather moderate, about 4, but also in some sediments of rather low pH, slightly less than 3. And these sequences were mostly related to Nitrososphaera venensis with high level of identity and with less low level of identity. And again, we basically know nothing about function of this Thermoarchaeota represented in acid mine drainage places. So what we know about whether there are some acidotolerant Taum Archaeota. There is a cluster of Taum Archaeota, which is Candidatus nitrosa-thalialis and genus Nitrosothalea, which were shown to grow at pHs between 4 and 6 and temperature between 20 and 40. And some of these organisms were actually shown to be in acidic soils and in other environments with rather acidic pHs. But again, they probably could be just acidotolerant, but not to be acidophile. So, and to conclude, what we could say that currently we know altogether 4 groups of Archaea inhabiting non-thermal IMD environment: Thermoplasmatalis, Microarchaeota, Parvoarchaealis, TMEC, and Taum Archaeota or Nitrosospheria. And Thermoplasmatalis are the most successful and studied Archaea in this environment, which could be represented as a major group. Cuniculiplasma is the most recent new genus described within this order, which is globally represented in IMD environments and also in some low temperature emplacement as well. And Cuniculiplasma could be also host for Microarchaeota. Their coexistence, at least under our conditions of cultivation, shows that they could coexist together, have common environmental strategy, probably, and some evolution, similar evolution history. And Cuniculiplasma also possess a certain adaptation for some period of time to very low temperature, and it could contribute to survival strategy for this organism. And TMEC and Taum Archaeota lack any culture representatives and need other further study. And of course, the 2 previous groups need further study as well. So, and at the end, I just would like to thank all people who actually participated in all this work, in all these publications. And also I would like to thank actually financing body, which is Royal Society, and also ERDF Thank you very much for your attention, and I'm happy to answer your questions.
Thank you very much, Olga. That was fantastic. It's fascinating to see such a deep dive into the phylogeny and ecology of the Archaea, the acidophilic Archaea, and it's so great to see like such a powerful, thorough approach to understanding, I guess, their evolution and their environmental distribution. I have some questions too, but I'm going to open it up to the group first. I'll save mine for last. So I'll open the floor to questions. You can throw your questions into the chat if you like, or please feel free to come back live and ask Olga your questions directly. Maybe I'll start with one if no one wants to take the first shot. Olga, I'm fascinated by the decreased genome size of the Archaea that you showed, and maybe this is just well understood and I've not been paying attention for a while to the Archaea, but is there a well-known environmental selective pressure in acidic environments to reduce genome size? Or, I mean, it's sort of a chicken and egg question, or is it just that reduced genome size somehow facilitates survival in such environments?
Yeah, so generally speaking, genomes of Archaea are smaller than genomes of bacteria. For example, if the biggest genome of bacteria is something about 15 megabase, for Archaea, I think Methanosarcina has the biggest genome, which is slightly more than 5. Speaking about acidophiles, acidophiles are generally have quite small genomes, acidophilic Archaea. And thermoplasmatalis basically have also quite small genomes, normally less than 2. Yeah, for example, Crane Archaeota like Sulfolobus, they have bigger genome, something about 3. Yeah, so it could be associated with their specialization to certain environments. They probably just, lost a lot of genes which they think they are not needed for them at all. Yeah, and just were exposed to certain selection to use just necessary genes. Yeah, it could be some environmental selection for them. And in case of, for example, Parvoarchaeota or Microarchaeota, they also lost a lot of genes and decided to rely on host. Probably it was less costly for them to live like this in environment.
Less energetically costly.
Probably, yes.
If I could follow up really quick, is that related to the utilization of the necromass, or is that a specialized function, or is that something that resulted from loss of functionality, do you think?
It could be actually, yeah, it could be both options. Yeah, Parvoarchaeota and Microarchaeota are actually also scavengers. They also rely on dead biomass. But probably some of them also have kind of, reduced capacity even in this. Yeah, they mostly rely on their host for this type of proteolytic machinery, I would say.
It's fascinating.
I was just going to ask you, are you able to elaborate any more on like the physiological adaptations that enable these guys to live at such extreme temperature and acidic conditions?
They actually are these conditions, which are very acidic, are considered normal for them. Yeah, because they are able to live just at very, very low pH conditions. They will die if they will be put at any neutral pH, right? And in terms of temperatures, they are inhabiting quite a big range of environments because, as I mentioned, they were actually also found in high Arctic environments where temperature is very low. And also in these places in Transbaikal area where very long freezing period is experienced by these organisms. but they are somehow adapted to this. In laboratory conditions, all thermoplasmatales are living at temperature normally 37, but they are also able, at least cuniculiplasma and ferroplasma, to live at rather low temperature of 5 degrees or 10 degrees. Yeah, so they are quite adaptable for environmental conditions.
Mm.
We have a question from the chat here. Did you find cobalamin synthesis genes or cysteine metabolic pathway genes in all of the acidic Archaea?
About cobalamin genes, I think that actually Parvoarchaeota and Microarchaeota probably will not have these genes. And in terms of cysteine, I think each organism needs to be changed because they have quite reduced capacity for synthesis of different amino acids. So when I mentioned cysteine, it was mentioned for actually kind of overexpressed proteins in cold conditions for cuniculiplasma.
And another good question coming in from the chat. Archaeota are involved in aerobic ammonia oxidation, and this process seems to be carried out better at circumneutral conditions. What do you know about ammonia oxidation by Archaea in acidic conditions?
I think we know nothing about this at all, because they were just relatively recently shown to be present as a minor group. And I think firstly, it was the paper of Carmen Falagan with her colleagues published in 2013. And we actually confirmed her data that Taum Archaeota could be present in this environment. And what we know is that there is some small amounts of ammonium in this environment, but it's basically, that's all what we know about this group. Yeah, I know that it could be quite actually crucial for nitrogen species in acidic conditions. But of course, certain efforts need to be done to isolate some Taum Archaeota from this environment, if it will be possible to do it.
Another question in from the chat here about the metal resistance genes that you showed. Were those mainly chromosomal, or is there a distribution across plasmids as well?
So they're mostly chromosomal. I think just probably some species of thermoplasma, I don't remember about Microphilus, have plasmids. Yeah, plasmids are pretty rare in this organism.
Interesting. Anyone else for anything else? Actually, I have a follow-up to Karen's question. She was talking about physiology. I was just wondering if the scavenging lifestyle leads to any sort of unique phenotype behaviors.
Phenotype behavior. Well,
whether it's morphological changes or biofilm preference, or
I don't think so. I don't think so. So they have no cell walls. Actually, why it's happened during the evolution process, it's also not very clear. Yeah, but well, somehow they are cell wall-like organisms. I don't think that it could be some connection with. But yeah, it's a good question. I probably need to think about this. And
well, there's too much to do. Clearly, that's fascinating. There's a rich amount of research to be done. Well, so I guess if are there any last questions that anyone wants to ask Olga in our formal session?
Hi, I have a question.
Please.
Sorry.
Thank you.
My name is Tomasa. Hi, pleased to meet you. My question is about Archaeota, and I found in some samples very high percentages of such Archaea in specific environments, underground environments near to the sea. And I had high percentages, like almost arriving at 8%, but I've only studied there at the level of 16S. So do you have any insight or information about like a relationship between their presence and maybe salinity?
No, not at all, not at all.
Okay.
What was the pH of this environment?
2.
Two.
Around
Yeah.
2. quite low,
Yeah, yeah, it's quite low. No, unfortunately, no. It's still very, very, you know, even not preliminary, just, you know, some indication of the presence
Yes.
of tau marchiota. Yeah.
Thank you very much.
Well, if anyone would like to stay behind for a bit and chat with the speaker, Olga, would that be okay if you've got a little bit of time? If anyone wants to stay for any
Yeah, sure, sure.
after seminar questions, it's sort of like the virtual equivalent of approaching the podium and having a little chat on the side. But otherwise, we will thank everyone again for joining us today. Please on, oh, I forgot the date now. Karen, remind me the date. Is it December 3rd? Yeah, she's shaking. Thank you. Sorry about that. December 3rd, we're looking forward to hearing from Dr. John Telling from Newcastle University, who'll be talking about the cryosphere microbiome as well. So thanks again to our speaker today, Olga, very much. We appreciate you making the time for us. And thanks everyone for coming out.
Thank you very much for your attention. Thank you. Thank you. So I'm still here, and
hi, Olga. Yep.
Yeah, is there any questions?
No, just to make sure that we did touch base with you from Kevin about recording, right? You were okay. Is that all right that we put your recorded talk up? Okay.
Yeah, it's all right.
That's fantastic. Great. We're trying to build up our bank of seminars.
Yeah, thank you.
Yeah, that's good. All right. You know, actually, I just wanted to ask you if you have a minute. I saw, I didn't realize. So I was in Jill's group at Berkeley when they were working on the Armon phyla, and I don't think I realized that they were phylogenetically closely clustered to peat bog species.
To
phyla that were coming out of peat bogs. I saw that in your
peat
tree.
bogs. Yeah.
I understand peat bogs can be fairly acidic. Is that right? Or
yeah, well, they could be moderately acidic, something I think about 4, maybe 5, yeah.
Yeah, how closely related do you think they might be?
They could be quite related, yeah, but the problem is that we need to be able to grow them in the lab and, you know, just to compare physiology between these groups, yeah, because currently we just basically could just make any kind of prediction just from metagenomics.
Well, that's true, absolutely. And so when you try to grow these things in the lab, is it fairly straightforward to follow some recipes and protocols, or is there some black magic involved in
absolutely, you know, absolutely not the case for this organism.
Yeah.
It's very hard to cultivate them, and microarchaeota are very hardly cultivatable organisms.
Yeah, that was my sense of it.
Yeah, and thermoplasmatalis as well, and about 2 other groups. Probably it will be some, you know, good progress for tau marchiota, because there are some people who is able to grow them, and tau marchiota, I think it's less tricky organisms in comparison to other archaea. At least it looks from, you know, my side. But about this TMEC, it's absolutely no information. Again, just predictions coming from metagenomic assemblies.
I wonder if you know my colleague, Zhenya Shelabolina. She was at Eric Roden's lab in Wisconsin for many years. And she formerly worked with Derek Lovely. When I first started working with Zhenya, I was very naive about cultivating or isolating extremophiles or even non-extremophiles, and I sort of, more with a geochemistry background, thought it was more about chemistry and recipes. But I very quickly learned that there's real artistry to it, and there's it's even somewhat, I even regard it as a bit of magic. I think it's sort of understanding, you know, how these cultures nuance and behave and have these symbiotic and synergistic requirements. It just seems so challenging. I absolutely respect people who are successful at cultivating and isolating things.
Yeah, yeah. There even, you know, for example, a colleague of mine, she has a background in medical microbiology, and she is always telling, oh, these environmental microbes are so tricky in comparison to normal ones.
Absolutely. Well, thanks again for making time for us this week. We really appreciate your seminar, and it was really, really interesting. Thank you.